Good morning, students. This is continuation to the last class about the research for safety and efficacy of herbal drugs, and in which we have seen about these protocols. We finished. We just made. I will continue about the guidelines, especially. Even I told about the 3 guidelines, which is very important and which is not present in your not detailed. They are not given in detail in your handout which you have, the printout which you are keeping, book which you are following. So this should be added. It's necessary for you to know about these things, and it should be added in your the chapters. Very important. And so that's why I added this one. And we can have this first one is, you can see the guidelines, especially the guidelines of quality specification of plant materials and preparation. Separately I have given, especially the quality of the specification for the plant materials and as well as products, especially the finished products which is available in the market, if suppose you are performing research, or even the products which is made in the research and development department in the pharmaceutical industries. Also the specifications, the guidelines we have to follow, what all the things have to be recorded before performing our research work. So in that, the second one, we can see the guidelines for pharmacodynamics and general pharmacological studies for herbal medicine and evaluating the safety and efficacy of herbal medicine. So we have to have another guidelines for pharmacological studies, therapeutic studies by animals, and even how we are performing before the clinical study. They have given some guidelines, what all the animals, and we are not going to the procedures, but at least we are giving the guidelines how and what type of animals and how we have to perform. And the third one, we can see about the guidelines of toxic investigation, that is toxicological study of herbal medicine. Toxicological study, I told about the toxicological study. You can see there the toxicological study, investigation of herbal medicine, which we have studied in toxicology and in pharmacology. We might have studied in the pharmacology 3. You have a chapter of toxicology, which is the scientist Paracelsus. He told dose makes the drug, whether it is a drug or it is a poison. That differs. For example, the dose, even water can become a poison, oxygen can become a poison. So dose makes the toxicity. So that's the Paracelsus, the scientist Paracelsus. You might have seen that there is acute toxicity, chronic toxicity, and you have subchronic toxicities, and we have many toxicity studies which we might have come across in drug. The same thing is applicable even for herbal drugs, herbal formulations, herbal materials. So we have made a guidelines, that is the guidelines which is given by WHO, and we have to follow the guidelines for quality specification of plant materials separately and plant preparations and formulations separately, and guidelines for pharmacodynamics. You made a pharmacodynamics, pharmacokinetics, pharmacodynamics, what drug does to the body and what the herbal drugs does to the human body, what it can be directly, we can, animal studies have to be performed in pharmacodynamic studies and general pharmacological studies of herbal medicine and evaluate the safety and efficacy of herbal medicine. And number 3, the guidelines of toxicity investigation of herbal medicine. As I told, toxicity studies is also very important. So these 3 studies we are going to discuss, and in this we will discuss only the 2, and the toxicity study, it was little wider, which we cannot finish today. With that we will discuss in the next class. So these 2 things which we can discuss, especially In the first one we will be having two specifications, one is for plant material, another is for preparations or the formulations. So in this slide, guidelines, first we are seeing about the plant materials and preparation, the quality specification for the plant materials and preparations. When you talk about all research on herbal medicine must specify the quality of the plant material or the preparation being investigated in order to study conducted by one investigator or by a collaborated by other investigator. One investigator, or we can have many investigators. You can see there will be a principal investigator and there will be many at all. You might have written at all in a paper when you are submitting. So there will be many investigators we are involved in a research can also be incorporated. So whatever may be, it is necessary for you to mention the quality specification of plant material or plant preparation which you have taken for your study. So this is an example which I have taken as an ashwagandha as an example, just I took from the internet and to show you. But what are the information? First we will see this ashwagandha. I have not given the full details, just an example, a picture I have mentioned there. And you see information, name and the characteristic of the plant material. This is for plant material. Preparation is separate. For plant material you have to write the name and specification like ashwagandha. What is the plant order? What is the family which belongs to Solanaceae? Withinia somniferum is the ashwagandha's botanical name. Withinia species, and Withinia is the genus, and somniferum is the somnifera is the species, and so that species also which should be mentioned, and the family botanical name should be mentioned, and the part which you are using. That's why the name of the plant material in Latin name, native languages, suppose if it is local languages, if it is available, that is also you should mention it. Not only from the native name and English, ever applicable, and the scientific name of the plant which reference to the author, and then family which it belongs to have to be mentioned, and the part of the plant used, in the condition also which should be mentioned. In what condition, dried or fresh, on what is the part, whether it is a dried rhizome or root, or it is sliced one, or it is for example scrubbing of the cortex or that is decorticated. These all should be mentioned very clearly. That's what we are mentioning there for you to have to mention very clearly in this part. And which part you are using, leaf or stem or bark or the root, and what type of this also mentioned. And when we did time, and the method of collection is also should be mentioned, it should be noted before in the research paper, and the preliminary preparation, drying, and if the material has been processed, if you are processing, that means post-processing, post-harvesting methods which you have studied, we have to mention that methods of processing, whether it is steamed or stir-backed or carbonized should be indicated. We have just a picture which is showed, that is a stir-backed beans. Just I have shown a picture that was stir-backed. So what type of processing you have made for the formulation, what type of processing you have made for the research purpose of the drug, crude drug, that is also should be mentioned. A brief description of the distribution and the habit of the plant growing wild or cultivated, whether you have collected from the wild or whether it is cultivated, include possible pesticides whichever it is used also it should be mentioned, documented properly the research. And if more than one plant species you are concerned for the research, and different should be indicated. If suppose you are using two species or two plants and combinations, so that differences should be indicated, and that sometimes the same species, same seeds, same pulses or same leaves of this different species, leaves can be used. So, but what is the difference specification? Photographs, drawings, sections, and all these things we have to, that is authentication, and pharmacognostical study, photographs, diagrams, and TS, LS, all these things should be specified. We are not mentioning all this here because already we have come across all the evaluation procedures we have seen in the pharmacognostical, and also for example we have seen in the previous chapters about evaluation of pharmacognostical evaluation and also botanical evaluations, chemical evaluations. These all things we have seen, so that we can incorporate here in between. That is a knowledge which you have, which have to be provided. And when we come to the characterization compounds, what do you mean by characterization? This is the chemical evaluation, and this is the various chemical evaluation. That's a one line which is given, but all things which we are having separate chapters also, applications of various instruments, applications of various chromatographic techniques, these all things which is included here. The characterization of compounds, the active principles of the compounds which is present in a plant, it should be characterized. How it can be characterized? How it can be extracted, isolated, and then characterized. There is a word which I am telling, that is three words: extracted, isolated, and characterization have been done. What is the meaning of that? Extraction, first you are extracting the material from the plant, and then we are isolating by means of chromatographic techniques, and we are separating the compounds, and compounds have to be characterized. The structure elucidation can be done by means of UV, IR, NMR, and mass spectroscopy, and even we are gas and mass spectroscopy, GCMS, and even emission spectroscopies, fluorimetries, which all this instrumental analysis which can give characterization and structural elucidation can be done, and you can prefer and you can specify the structural activity relationship which can be useful for the pharmacological and therapeutical activity. That is what characterization compound of the plant material, which may be the biological and therapeutical activity principle of the particular compound or the particular plant which is present, should be quantified, assayed and determined, and their structural formula, particularly if they are uncommon, they should be specified, they should be researched, and it should be identified, and the process of plant material changes in the quantity of these characterization compounds should be described in your research. So that's what we are making here very clearly, this paragraph, and it has many type of chapters which comes in the future, and as well as which you have studied before also in analysis and in various subjects. So these knowledges can be incorporated here by means of the students to just what I am saying here. And then quality specification of the material. This is talking about the plant material. Quality specification, I just put only the authenticity, purity, assay, packing, labeling, and storage. But don't think that only these points is needed. The point should be elaborated by the student's knowledge, especially authenticity. What do you mean by authenticity, authentication? There are many institutions, and the qualification are a botanist who is having the PhD in the botany, and he should be certified. And it should be certified by a cognoscist or by means of botanist. He has to certify that his botanical name and the selected plant material which is exactly that particular species and belongs to the family, and the floral diagrams have to be mentioned, and it should be herbarium have to be preserved, and that is called authenticity. That is specified authorities are there in each and every countries will have that. Authenticity is very important before a research. Purity of the compound, and adulteration or additional of anything, purity of the compound have to be determined by means of assay, determined by means of various sectors, various methods of preparations, procedures are there which has to be followed for the purity. That we have to mention how you are going to find out the purity of materials of herbal. And assay, it is determination of amount of the substance. When you are talking about the secondary metabolites or the compound which is responsible for the therapeutic activity or the pharmacological activity, assay, determination of amount of alkaloids, determination of amount of certain chemicals, specific chemicals like tropane alkaloids, like atropine, or any type of compounds can also be mentioned clearly about the assay compounds. Packing, labeling, and storage. What type of packing you have received, what type of packing and labeling have been made already. If you are taking a product already. Sometimes we will take plant materials which is available in the market. There are leaves available, leaf powder is available, root powder is available, stem powder is available, bark powder is available. There are many types of powders and many type of crude drugs also available, which is mentioned for the usage of the direct patients. So that's also we should mention about the packing, labeling and storage. So this is about the herbal materials. When you want to go for a research purpose, these are all the points which is necessary for the quality specification of the compound. And next step, when you come to this is about the preparations or the formulated products, preparation of the plant material. That is only plant material. Information for the medicinal preparation of the plant material. That is one is only plant material. Preparations of plant material means we mean that formulated and it is extracted material or a compound which is isolated material which is available in the market in the form of various formulation, in the crude form, not in the crude form, it is isolated form, and maybe the formulation may be a traditional method of formulation when a capsule, tablet, injections and even a syrup form which can be given isolated compound which are prepared in the formula. So that preparations, what all the materials which you have to, what all the specification which you have to mention, what all the things, the guidelines says, what is the things which, before research, what all the things you have to mention. So you can see here among the medicinal preparation, there are widely used powders, granules, pills, extracts, tablets and injections. These all are even herbal medicines we have. So these all products, when you are taking for the research, it is necessary traditional powder and pills are made into powder, plant material, tablet, granules, ointments and newer types of pills, mostly made into extracts which is available. Injections are also made purified extract and pure active constituents isolated from the plant materials are also available. So these all comes in this. And what is the information, what is the guideline says? There are also certain medicinal preparation made for both powder and material extracts also available, which I have not mentioned. Even this is also present. So there are external preparation ointments, creams and even powders and isolated compounds which is in the form of, even in the injection form, tablet, capsule, syrups and all the various pills and granules are also available. So these all compounds, what are the things you have to specify? Name of the formula first and the product. Name in the Latin name, English name and in the native names which is available, what is the product which is available, it is also which should be mentioned as how you mention in the plant material. Same thing have to be followed. And formula should be indicated whether it is ml or in the grams. That is what the name and such ingredients, sometimes combination, quantity used for 1000 grams or 1000 ml of the product, if it is solution or a liquid, 1000 ml, it should be mentioned. And the excipients which are added. Suppose you are adding diluents, suppose you are adding an starch or adding a binder or a coloring agent or a coating agent, or sometimes if you are adding a lubricant, all the excipients, whatever you are doing for formulating, it should be mentioned, specified very clearly in the label. Understand, that is what even in the research. And the method of preparation, how they prepared, that is also very important. Before you are going to research or to find out the safetyness, or you are going to identify the animal study, or you are going to pharmacological study or toxical study, or you are going to whatever the study which you are going to study, or extra formulating, new formulating, that is purpose of the study which also we have studied there. In the last class we told what is the research purpose of the study. The purpose of the study may be new formulation, maybe new identification, no indication, new combination of the study. There are many, or evaluation, and also it can be done for even safety and efficacy of the therapeutic activity already existing. Whatever may be the research topic which you have taken, but it's necessary the method of preparation, how you have taken the preparation is very important. So 100 grams and 100 ml of the product and description of the method should be included, details of the process such as solvent which is used, and also the temperature which is used for extraction, and the concentration, what type of temperature you are used for concentration, as well as process which is used and reduced to the level of the microbial contamination, whether you have used any pasteurization method or by means of any type of sterilization agents we have used, or you have used for a hot hot, what type of moist heat type of sterilization, or you are trying dry heat type of sterilization. Sterilization, lot of procedures you might have studied. So what type of method you have used for removing microorganisms and to preserve them, and what type of preservatives, that all I have not added. All details when you are going for a product, when you are going to take a product, or when you are taking a material preparation of a herbal preparations for any type of specification, it is necessary for you to detail of the procedures. And active constituents should be stated with the structural formula, very important, structure. If it is an active constituent, it is necessary for you to know about the structure of the formula given and the chemical or the pharmacological incompatibility, the chemicals which is incompatibility with the other compounds. So that is what a cautions. We cannot take, this should not be taken along with these certain compounds, it should not be taken along with certain foods. So these all things should be mentioned, the incompatibility, pharmacological incompatibility should be mentioned. And in the research with topic which you are taking a sample. This is a picture just to show you about the sample of a product which you have taken. But there are many products, whichever the product, whether it, I have just shown a liquid and a solid preparations. Any preparation, sometimes it can be an injection, it can be a tablet, it can be a syrup, it can be a capsule, it can be even nowadays novel drug delivery systems also, it can taken as a formula which has been taken as a from a herbal medicinal products. So now we are quality specification, again I specified what I specified in the material, the same thing I have specified for the preparation also. Herbal preparation also we are mentioning that authenticity, purity, assay, packing, labeling and storing. So it is not possible for the students to just to write only the line of authenticity if suppose they are asking any question. Authenticity means you should explain what do you mean by authentication. How to authenticate, what is the qualification, who can authenticate the herbal drugs, who can authenticate the herbal formulation. So there should be some chemist, pharmacist which is responsible for authenticity, for authenticate the product. This is the product which is prepared from this formula, and this is the product which is prepared, this is the and also if you are telling about any type of material or any type of plant material, it is necessary for you to identify the botanicity of the particular compound, and chemistry should be identified, authenticity with an authority, and also what all the list of institution responsible. Even in India we have given some list of authority which is responsible for authenticity, which can be received for any type of researcher before starting the research. So purity, as I told, purity is very important to know about. It is the adulteration, collection, and what type of collection it is made, what type of adulteration can be made. This also included even any type of pesticides, any type of debris, any type of added, this all. And as I told, suppose constituents, when you are talking about the constituents, it is necessary for you to know about the assay determination. A determination of amount, and the procedures have to be mentioned, whether it is an alkaloid, whether it is a specific alkaloid, or whether it is a glycoside, a specific digoxin, or any type of A glycoside or B glycoside or C glycosides, so whatever the glycosides, whichever specifically how we can identify what is the procedures of determination have to be mentioned. And packing, labeling and storing, as usual, how the packing is made, whether it is a blister packing or whether it is a packed in a bottle, or whether what type of things, and labeling, how the label is mentioned, and storage conditions and what type of conditions is needed. All these things should be mentioned before research is going on. So the two things we have seen, but the same slide which has been repeated for you for plant material and plant preparation. That means plant preparation, herbal plant preparation means herbal formulations. So these both of them you need. What all the quality specification is similar. And now we are coming to non-clinical studies. Even non-clinical study, I have made one slide for you to think about non-clinical studies. The primary objective of non-clinical studies are to determine whether such studies support the clinical study of the herbal medicine. To determine whether such studies, which you are going for a studies of a research study, support the clinical use of herbal medicine. So you should know about whether this herb is having any clinical use, whether it is having any therapeutic identity. So these can be easily identified by means of various literature survey, and also you should know, specify very in a non-clinical study. And in non-clinical study, the range of pharmacological action of herbal medicine which can give characterize how How pharmacological study is a clinical, for example, non-clinical study. In animal study we can make, and we are not going for the clinical study, we are going to study about the pharmacological action. With the animal studies can also be characterized by the chemical constituents which you have isolated, and this also can be determined. And number 3, it can define the chemical characteristic of pharmacological activity. For example, when you are identified by the various instrumental analysis and identified the structural isolated and characterized chemical compound, and if it having specific pharmacological activity in the animal study, so that you can just see the characterization, or we can see the pharmacological. So because you might have studied in your medicinal chemistry and even pharmacology, there will be a pharmacological type of classification, and also there will be a chemical classification. They both will be having a relationship, and when you study we can find out there is a relationship of the chemical classification and the pharmacological classification, which is having relationship with the structural activity relationship which you are studying in the medicinal chemistry. So that's what we want to see, that the chemical characterization of the pharmacological active natural products, and to elucidate the mechanism of action. Mechanism of action. What is the mechanism of action? Pharmacological mechanism of action can be easily identified, and also we can make in non-clinical studies. Clinical study this separately in this chapter only. Finally we will discuss. So now we are coming to discuss about the guidelines of pharmacodynamics and the general pharmacological study of herbal medicine and pharmacodynamic investigations. So in that, animal studies of how we are performing. So pharmacodynamic investigation are conducted in the light of the expected therapeutic effect of herbal medicine using appropriate non-human system. And general investigation of general pharmacological investigation. The pharmacological investigation are conducted to elucidate the various pharmacological activities other than main pharmacodynamic action. So apart from that main pharmacodynamic action, which is actually such investigation usually over the test of nervous, necessary cardiovascular, respiratory system, and if necessary even other systems can also be examined, should be performed on conscious animal, instead of saying unconscious, anesthetized animal, using adequate doses, low, increasing doses, high doses, using even proper route of administration, different routes of administration, like you can see intraperitoneal, intramuscular, even oral, and even external applications. These all things have to be identified. So intraperitoneal, because vein, it is intraperitoneal route. You might have studied about intraperitoneal route, how we are maintained. So route of administration also plays a major role. Intraperitoneal route, which is equivalent to the intravenous. So which can animal, which we can perform the intraperitoneal region, which is near to the abdominal region, which can be injected, which your pharmacology teacher might have told you how to take the intraperitoneal injection. So herbal medicine have various pharmacological effects. First, how the animal, I am not going to the detail about the pharmacological procedures of any examples or anything, but just animals which we can perform for the herbal pharmacological effect study. So species, appropriate animals including mice, that is albino mice, albino rats, guinea pigs, rabbits, cats, dogs, etc. can be used. Even goat can also be used. And you can see characteristic of the animal, such as strains, as I told, albino, rats, and sex, female or male, how many which you have to celebrate, all these details there will be procedures will be given. Age factor, and also holding the condition should be also specified, and also even weight of the body weight or the weight of the animal is also specified, should be specified, which I did not mention there. So animals like you can see use rabbits, mice, guinea pigs, and or very commonly guinea pigs are very useful for antihistaminic action, which you are always using sometimes in the pharmacological action, which you might have studied the acetylcholine action, which you are seeing by ileum, which you are seeing, I hope. But when we are studying, we were using animal study. Nowadays, due to animal sacrification should be reduced, and it is not possible for you to do without permission. So that's why you are always doing by means of computerized pharmacological study which you are making. Disease models can be made by treating animals with certain chemicals and other modalities. So you can see certain chemicals. For example, cancer. If you want to see the any type of anti-cancer drugs, if you want to, you have to induce cancer by means of aloxone. Here, immunological study. Here I have given an example for immunological study. Using cyclophosphamide induce the immunosuppressants, and you can find out the drug which you are using or the plant which you are using or the extract which you are using, whether it is having immunostimulant effect, can be seen in the animal. So example, immunological depression mice can be made treating with an immunological suppressants like such as cyclophosphamide. Such animals can be used to evaluate immunostimulants activity to the test of medicine. Just an example I have given. You may be knowing more than me. You can write many examples. And here, for genetical defects, animals with genetical defects can be also be useful. We can have bone marrow, kill the bone marrow of the animal, and then we can implant the thymus cells in the tissue, liver cells, and induce the human stem cells, and which can be studied in the rats, in the albino rats, which I have shown in the picture. You can see that it is also possible for you to study. And autoimmune mouse. Autoimmune disease, very common disease. You might have seen autoimmune is a common diseases in the certain ages. Usually people are getting arthritis and many type of, even you might have studied many type of diseases, and this hypertensive animals can also be studied. Those herbal medicines can be used for under principle of traditional medicine. Animal models needed to establish according to those principles can be used. And we can use whole animal in certain experiments, because I am not giving the specific, only the name I am giving. Whole animals can be used. There are many experiments, many examples. Whichever you know, you can write. If they are asking something, essay question like pharmacological activity study in a research, how will you do for an herbal medicine? You can write. Whole animals can be used. Isolated organ of a tissue can be used. A kidney can be isolated. A liver can be isolated, and you can identify isolated organ, even especially isolated organ when we are using frog for heart, and isolated organ and tissues can be used, and blood samples and the components can be studied from the animal, and a tissue, that is ex vivo studies, but tissue studies, cultural studies, and tissue culture studies can be also, tissue cells can be studied, and subcellular constituents study also can be done. These all are some of the tissue culture studies, and tissues in the animal tissue studies and the plant tissue studies can also be added and performed in this animal pharmacological studies. It can be included. Administration, when we talk about administration, route of administration. Since oral route is very much useful and very much easy form in the herbal formulation and herbal medicine, usually they are used clinically. The oral route of administration is the most suitable route, as we have used. Usually you used to use some catheter and then a syringe, and then we will just give the oral route for the animal as shown in the picture actually. But you might not have experienced this one by means of, because you are not done any experiments. So usually by means of using a catheter, and it is possible, and there will be a wooden piece which can be kept in, because otherwise the rat will bite you. So you can keep there, and it is possible for you to give the route of administration by oral. And not only the oral, sometimes if we need it, we have to go for other route of administration also. Usually doses are selected for a study should be established through the dose-response relationship. Dose-response relationship curve you may be studied in the pharmacology, and the selection of the dose of the animal study should be accordance with the regular clinical doses. Regular clinical doses. Suppose if the clinical dose is given in the parenteral route and you are not able to see in the oral route, so whatever the route which is specified, whatever the route, external means external, internal means internal, parenteral means parenteral, oral means oral. So that should be studied even in the animals. Control group always you should have. what is a solvent system, that should be given a control group of animals you have to make without drug. So a study must be included negative, vehicle only which should be given control group of animals have to be studied to have a comparison. And now the next class we will continue with the guidelines of toxicity investigation of medicine, because toxicology and toxicity investigation is another is the third one which we want to discuss, because we have discussed about the materials and the formula and the preparations, and then we have discussed about the pharmacological. Simply we have given some examples of animals which we can use for a pharmacological study. And now we have to go for toxicological study, which is very important for the safety and efficacy of herbal drugs, which we will discuss in the next class. Thank you for being with me.